Please try to reconnect. Okay, the record has started. Okay, so what I was saying that usually, I mean, we used to take the mid within the class hour during the midweek. So, for example, the midweek was 21 to 26. So, we were supposed to take the mid on 22 or 24 November. Okay. But you know that this is not a normal situation, so we can also act abnormally. Okay, let's not fix the date today. Remind me in the next week, but please keep it in your mind that maybe we'll take the mid on 24. Okay, 24. Is 24 okay for everyone? I mean, if we take the mid on 20, just a minute, we have a class on 24. Yeah, 24. Is it okay for you?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir. No
Yes,
problem.
sir.
So, anyone have any
Okay,
other
sir.
mid on that day?
Sir, we haven't decided our other midterms yet, so
Okay. Okay. So let's fix the. So what happened? Like this is a single section course. So setting a date for this course is not that difficult. But for multi-sectional course, they sometimes. I mean, it sometimes become complex to set a fixed date. So I guess you have only one multi-sectional course, maybe Math 091 or English 102. So let's fix 24 as a tentative midday. But if you have another mid on that day, please let me know. Okay.
Okay, sir.
So
Okay, sir.
Yes,
you are
sir.
a fresher and you are in the first semester, so I don't want that you, I mean, you sit for 2 mid at the same day. I don't want that at all. So if anything happened like that, I mean, you have another exam on 24. So in that case, we can reschedule the exam date. Okay. And about mid, the mid is okay. Just first I'll keep the record. MIC 101 mid. The mid will be almost same like the quiz. The only difference will be there will be more question and the mark will be 30 mark. It will be a 30 mark exam. There will be more question and you will get more time to finish the exam, like around 10 to 12 hours. Okay.
Okay, sir.
Okay,
I
sir.
haven't checked your quiz script yet, so I cannot tell you the mark today. So why are you okay with the question pattern? Any difficulty in answering or anything you want to share, or you have any comment?
No, sir.
It was fine, I guess.
It was a good question.
Okay, then it's fine. I mean, it's an open book exam, so you can take information from anywhere, but your answer should be, I mean, should be consistent. I mean, not consistent. It should be pertinent. I mean, it should be go with the question. Because what several students do, they just look for the information in the net and copy and paste the whole thing. Okay, you can take the information from the net, but it doesn't mean that everything you get in the net will be the part of the answer. Okay, so please answer carefully. Okay, so we discussed until which one? Lecture 3. Lecture 3 is done, right?
Yes, sir.
Okay. Huh? Didn't we discuss about lecture 3? I think so. Hey, lecture 3 আমরা lecture 3 নিয়ে কথাবার্তা বলে ফেলছিলাম না?
Yes, sir. Yes, sir.
Okay. So okay, guys. So we need to speed up. Okay. So we have several lectures left. So we need to speed up. Okay. So please, I mean the video lectures are already there. So please look into those. I mean, watch those lectures and ask questions during this online session. If we cannot speed up, we'll lag behind. একটু 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 আগাতে হবে, হ্যাঁ, পড়াশোনা একটু আগাতে হবে, না হলে আসলে আমরা পিছিয়ে পড়ে যাব। বেশ কিছু লেকচার বাকি, তো এখন যদি আমরা একটু আগায় না থাকি, শেষের দিকে বেশ একটা পড়ে যায়, প্রেসার পড়ে যায়, এটা করা যাবে না। Okay, so anyone have any question from the next lectures, like lecture 4 or lecture 5?
স্যার এই ভ্যাকসিন জিনিসটা আসলে কে ইনোভেট করছে? কারণ we see that Edward Jenner is the one who made the first one with his cowpox one, but then Louis Pasteur took the spinal cord and brain from rabbits, pulverized that, made the glycerin, and then made rabies vaccine. So who made the first vaccine? Like, who do we give the credit to?
If you say, if you ask the question that way, then I would say Louis Pasteur. Edward Jenner was the first scientist who was able to give an idea about the vaccine thing. So if you look in his work, you can see that he didn't clearly mention that what will give the protection, which part will work as the vaccine, right? He just said that if people get exposed by another virus, they get protection from another virus. It was like that, right? If people get exposed by cowpox, they get protection from human pox. Okay, and in his approach, he didn't isolated the pathogen. He didn't isolate the virus or bacteria. He kind of used the whole component. Okay, so it's like, say for example, উনি যেটা আসলে করেছিল, উনি ধরো কেউ যখন ইনফেক্টেড হয় বা এক্সপোজড হয়, সো তার কাছ থেকে ধরো অনেকটা আমরা এটাকে সেল লাইজেট বলতে পারি, অথবা একটা ধরো, say for example, someone got infected and you took collected sample from that person, and you re-injected it into a healthy person, and the healthy person get protected. Okay, that means immunity was induced in his body. So he took the whole sample. He didn't isolated the specific pathogen. Okay, he knew that the causative pathogen were present in that sample. He knew that. Okay, but he was not able to do the pure
Isolate
culture. He was not able to isolate the pathogen. Okay, a specific pathogen.
Like using Koch's postulates that Pasteur did.
Yeah, something like that. মানে উনি কিন্তু একদম পিওর। নরমালি এখন ভ্যাকসিনে আমরা কি ইউজ করি? আচ্ছা, ভ্যাকসিন বিভিন্ন ধরনের হয়। হ্যাঁ, ভ্যাকসিনে আমরা পুরো মাইক্রো অর্গানিজম ইউজ করতে পারি, আমরা এটা একটা পার্ট ইউজ করতে পারি। ওকে, সো অ্যাজ এ ভ্যাকসিন উই ক্যান ইউজ দ্য হোল মাইক্রো অর্গানিজম or we can use part of it. Okay, but in either way, first we need to isolate that microorganism. We have to make pure culture of that microorganism, then we can use that as a vaccine. Okay, so Pasteur was more how can I say, his approaches were more specific. Okay, but usually I think scientific society, they regard Edward Jenner as the so-called father of the vaccination thing. Okay, but anyway, it doesn't matter at all, right?
Yes, sir.
Someone invented it, so it's okay. Okay, so if you don't have any further question, I'll just give an overall idea about lecture 4 and lecture 5 today.
Sir, lecture 3, I pitched question to you.
Yeah, please. Yeah, you can ask. Of course, you can ask.
So chemotherapy, for a criteria of a good antimicrobial chemotherapeutic, should not allow development of resistance in the pathogen during time of treatment. Isn't it possible that the pathogen itself can develop resistance? And it's not always guaranteed that a chemotherapeutic reagent will not allow the pathogen to develop resistance, right?
আচ্ছা, ওখানে কিন্তু ওই কথাটাই বলা হয়েছে, মানে কেমোথেরাপিউটিক এজেন্টটা এমন হওয়া উচিত না যাতে প্যাথোজেনটা ইজিলি রেজিস্ট্যান্স গ্রো করতে পারে। এই সেকেন্ড 2 নাম্বার পয়েন্টটা না,
জি স্যার।
should not allow development of resistance in the pathogen during the time of treatment. এখানে না ওই কথাটাই বলা হয়েছে যে নরমালি যেটা হয় কি, এটা সব যে কোনো বায়োলজিক্যাল সিস্টেমেরই স্বভাব হচ্ছে তারা যখনই কোনো বাধা পাবে তার এগেনস্টই তারা একটা ইমিউনিটি মানে একটা প্রোটেকশন গ্রো করার চেষ্টা করে, রেজিস্ট্যান্স গ্রো করার চেষ্টা করে। এটা বায়োলজিক্যাল সিস্টেমের স্বভাব। সো কেমোথেরাপি কি? কেমোথেরাপি হচ্ছে কেমিক্যাল। সো যখন এটাকে আমরা ড্রাগ হিসেবে ইউজ করতেছি মাইক্রো অর্গানিজমের এগেনস্টে, সো মাইক্রো অর্গানিজম চেষ্টা করবি, এটা বায়োলজিক্যাল সিস্টেমের ইনহেরিটেড বৈশিষ্ট্য আর কি। হ্যাঁ, তারা চেষ্টা করে একটা রেজিস্ট্যান্স গ্রো করার আর কি। সো তারা রেজিস্ট্যান্স গ্রো করার চেষ্টা করে। সো কিন্তু কিছু কেমিক্যালের এগেনস্টে হয়তোবা তারা মোর ইজিলি, আই মিন দে ক্যান গ্রো দ্য রেজিস্ট্যান্স মোর ইজিলি এগেনস্ট সাম chemicals. So those chemicals will not be suitable to use as medicine or drug. ঠিক আছে, যে কেমিক্যাল গুলোর এগেনস্টে সহজেই রেজিস্ট্যান্স গ্রো করতে পারে মাইক্রো অর্গানিজম গুলো, সেগুলোকে আমাদের ড্রাগ হিসেবে ইউজ করা উচিত না। মানে সেটা একটা ধরো ভালো কেমোথেরাপি, অ্যান্টিমাইক্রোবিয়াল কেমোথেরাপিটিক না।
So sir, how do we develop a type of chemotherapeutic agent that does not allow development? মানে second criteria টা fulfill করার জন্য how do we have to design the chemotherapy?
যেটা হয় যে it's just we have to do the trial, we have to check. So what happened, like say, okay, so if we say scientifically the steps are like this, we try to know the mechanisms of the life cycle. I mean, how say it's a bacteria, so you want to discover or develop a chemotherapeutic against a bacteria. So we should know about the bacteria. Okay, what is the structure of that bacteria, how that bacteria multiply in our body. Okay, how it do the pathogenesis. Okay, so say for example we got infected by a bacteria, so first it needs to enter in our body, right, through its preferable route of entry. Then after entry it have to colonize the target cells. Say for example that bacteria is a respiratory pathogen, that means it infect our respiratory tract, lung. So first it need to get into our lung and then get attached to the lung tissue, and then it have to enter into the lung cell and multiply there. Okay, so if we know their mechanism, then we can choose the chemical. So this chemical can target, I mean, this whole pathogenesis, it has several stepwise events, right? It's entering in our body and getting attached in the tissue, then entering into the tissue, then multiplying there. So we have to choose a chemical which can prevent any of this step. So for example, you find a chemical which prevent that bacteria to get attached with the tissue, tissue সাথে attach হতে দেয় না, then it can act as a drug. Or say that bacteria, it get attached with the tissue, then to do cause the disease it need to multiply. Say you find a chemical which can prevent this multiplication. So if the bacteria cannot multiply, they will not be able to increase their number, so they will die eventually. So it will again work as a, I mean, then you can use that chemical as a drug. So did you understand my point?
Yes sir, thank you sir.
না বুঝলে আমাকে বললো, হ্যাঁ।
না স্যার, বুঝছি, thank you.
So however, in reality the things is different. Okay, what I just explained or discussed, it is like a by book thing. Okay, but in reality what happens, the people, I mean the scientists who work with drugs, I mean who try to develop new drugs, they have several chemicals in their library, like 1 lakh, okay, 50,000, 100,000, 150,000, even 200,000. They have different chemicals, okay, in their collection. So they just try the chemical against the bacteria and then analyze which one give the best result. এরকম না অসংখ্য কেমিক্যাল আছে সায়েন্টিস্টদের কাছে যারা ড্রাগ নিয়ে কাজ করে, তারা এরকম বিভিন্ন ড্রাগ গুলো না ট্রাই করতে থাকে। ওই প্যাথোজেনের উপর। ধরো কোনো একটা ব্যাকটেরিয়া, ওই ব্যাকটেরিয়াটাকে এগেনস্টে তুমি একটা ড্রাগ চাও। সো বিভিন্ন কেমিক্যাল তারা ট্রাই করতে থাকে এবং দেখে যে কোন কেমিক্যাল গুলা কাজ করে। তো যেটা কাজ করে তখন সেটা নিয়ে আরো না ফার্দার ট্রায়াল হতে হয়। হ্যাঁ, একটা ড্রাগ ডেভেলপ করা, এটা আই মিন ইট টেক লং টাইম টু ডেভেলপ এ ড্রাগ। ইট টেক ইউজুয়ালি at least 7 years. Okay, at least 7 years. Usually it may take like 5 to 10 years on average for a drug to discover and bring it in the market. It takes long time. Okay, because say for example, I mean the first research you have to do in the laboratory, and then in the laboratory if you find a potential chemical, then you have to go for trial. Say you find a very good chemical in the lab, it doesn't mean that you can use that as a drug. Okay, because there are safety issues. So first you need to do the trial to check whether it will be safe for human use, whether it will be really effective for human, because you just discovered it in the laboratory, not in the real human body. So trial is necessary, and trial has there are two types of trial. Preclinical trial, it is done on animal, and then if it is safe in the animal, then we can go for human trial. যে কোনো কেমিক্যাল ইনভেন্ট করার পর ট্রায়াল করতে হয়। ট্রায়াল খুব নেসেসারি। ট্রায়াল ছাড়া আমরা কোনো কিছু মার্কেটে আনতে পারবো না, কারণ হিউম্যান খুব সেন্সিটিভ জিনিস, সেফটি খুব বড় কনসার্ন। এরকম অনেক কেমিক্যাল না আছে যে কেমিক্যাল গুলা খুব ভালো অ্যান্টিব্যাকটেরিয়াল ইফেক্ট দেয়, কিন্তু সেগুলো হিউম্যানের জন্য সেফ না, এই জন্য ওগুলোকে ড্রাগ হিসেবে ইউজ করা যায় না। আচ্ছা, দেন ওকে, দেয়ার ইজ অ্যানাদার পয়েন্ট. Okay, don't worry, you will learn these things. I mean, you are just in the first semester, so throughout your journey in microbiology, you'll learn about all these things. So, like, say for example, a new bacteria has been discovered, and it's a novel bacteria have been discovered, and you want to find a drug for it, antibiotic for it. So you just don't need to try randomly several thousand chemicals. Why? Because when you find a new bacteria, a novel bacteria, first you will try to identify it in which groups it belong to. If it belongs to a already known group, then you actually already know that which type of drug can work against it, right? So in that case, you have to search the antibiotic in limited number of chemicals. You don't have to go, try with all the 50,000 or 100,000 chemicals you have. ধরো তুমি একটা নতুন ব্যাকটেরিয়া পেলা, হ্যাঁ, প্রথমে কি আমরা তো সেই ব্যাকটেরিয়াটাকে আইডেন্টিফাই করার চেষ্টা করি। তা আমরা প্রথমেই চেষ্টা করি কি যে এই নতুন ব্যাকটেরিয়াটা কোন গ্রুপের ব্যাকটেরিয়ার মধ্যে পড়ে। সো অলরেডি তো মার্কেটে অনেক ধরনের কেমিক্যাল আছে যেগুলো ড্রাগ হিসেবে ইউজ করে। সো আমরা যদি আইডেন্টিফাই করতে পারি আমাদের নতুন ব্যাকটেরিয়াটা কোন নন গ্রুপের মধ্যে, জানা গ্রুপের মধ্যে পড়ে, তখন আমরা ওই কেমিক্যালগুলো ইউজ করবো যেগুলো ওই জানা গ্রুপের এগেনস্টে কাজ করে। পয়েন্টটা বুঝতে পারছো?
জি স্যার।
Okay, so any more question from lecture 3?
No, sir.
Okay, so if you don't have any more question from lecture 3, then I will just give you a brief about lecture 4 first. Lecture 4. Okay, so lecture 4 is a very easy lecture, and there is nothing complex which I should explain. It's just mention some of the use of microbiology for human welfare. This I would say this PowerPoint slide is like it's a poor PowerPoint slide. Okay, I mean there are not so many information there. If you look the Tortora, you have the Tortora microbiology book, right? I have shared it in the classroom. Am I right?
Yes, sir.
Okay.
Yes, sir.
So if you look into that book, and you will find a similar chapter there. I mean, like use of microorganism or microbiology in human welfare. You will see lots of different uses of microbiology for the human welfare. Okay, so this chapter is about that. This chapter just, I mean, in this PowerPoint slide, there are only few use of microorganisms. So we have a common misunderstanding that microorganisms mean bad. Okay, they do disease, but only few numbers of microorganisms cause diseases. Most of the microorganisms, and there are many microorganisms which are beneficial for us, which don't do any harm to us. Rather, they do lots of benefits for us. Okay, we human use many microbes for our benefit in industrial, I mean in food sector, in industry, for environment control, in agriculture, okay, in health and sanitation, okay, pollution control, medicine, as medicine therapy, okay. So that's mean if you are in microbiology, I mean you are in microbiology, so it doesn't mean that you all have to work with the disease. So you can see, just, I mean, although these PowerPoint slides are poor, but from here you can see there are many use of microorganisms in different fields. So in future, you can choose any of this field and work there. Microbiology technically এটা একটা broad subject, হ্যাঁ, যে মাইক্রোঅর্গানিজম মানে অসুখ বিসুখ এই ধারণা থেকে আমাদের বের হয়ে আসতে হবে। শুধু অল্প কিছু মাইক্রোঅর্গানিজমই আসলে ডিজিজ করে, বেশিরভাগই হচ্ছে হেল্পফুল। হ্যাঁ, আমরা অনেক মাইক্রোঅর্গানিজমই হচ্ছে কি জানি বলে, মানে ইউজ করি কিন্তু আমাদের বেনিফিটের জন্য। আমরা অনেক ইন্ডাস্ট্রিয়াল প্রোডাক্ট আছে যেগুলো মাইক্রোঅর্গানিজম দিয়ে আমরা তৈরি করি, তা মানে ব্যবহার করে আমরা প্রডিউস করি। তারপর হচ্ছে অনেক মাইক্রোঅর্গানিজম আমরা হচ্ছে থেরাপিউটিক বা মেডিসিন প্রস্তুত উৎপাদন করতে তৈরি করি। ওকে, এগ্রিকালচারে, মানে এগ্রিকালচারে ইউজ হয়, তোমার এনভায়রনমেন্ট পলিউশন কন্ট্রোলে ইউজ হয়, ঠিক আছে, স্যানিটেশন, যেমন ধরো সুয়েজ আছে না, সুয়েজ ট্রিটমেন্টে ইউজ হয়। ওকে, দে আর many uses. Okay, so scope, lecture 4, scope of microbiology in human welfare. I'll just give you some examples. So for example, food and beverage. In bread industry, for bread making we use Saccharomyces cerevisiae. It's the yeast. Okay, it's not a bacteria, it's the yeast. Okay, by the way, you all know how to write the scientific name, right? So Saccharomyces cerevisiae.
Yes, sir.
So either we have to write it in italic form, and in this case this letter will be capital. This is the species, and this is the this is the genus, right? Genus, the first letter of genus will be capital, and the first letter of species will be in lowercase. Okay, and either
Yes.
we have to write it in italic form or in bold font, or we have to put a underline. So usually when we write, I mean do handwriting, it's difficult to write in italic way, right? So in that case we just write the name and put a underline. Okay, so you all know that, but still, I mean, sometimes students do this mistake while they write their answer scripts, so please take care of it. These are silly mistakes, but don't try to, I mean, just try to avoid those kind of mistakes. I mean, they looks bad in the script. Anyway, so let's go back to the lecture. In bread industry, Saccharomyces, yeah.
Do we need to memorize the names of the microorganisms?
Not all. Okay, so eventually you will memorize many of the name. Okay, I mean, you don't have to give extra effort to memorize this name. Eventually you can, I mean, you will able to memorize many name of many microbes. আমরা যখন একই ফিল্ডে অনেকদিন ধরে থাকি না, এমনি না মুখস্থ হয়ে যায়। এই মাইক্রোঅর্গানিজমের নামও এমনি মুখস্থ হয়ে যাবে অত এক্সট্রা এফোর্ট দেওয়ার দরকার নেই, ঠিক আছে? Okay, so in bread industry, Saccharomyces cerevisiae is used for cheese making, yogurt production, doi, sauces, coffee, alcoholic beverage, a very big industry, and alcohol are produced using microbes. I mean, from where we produce alcohol? From raw material. Like we can use very different type of raw material, say grape. From grape we can produce wine, rice. We can use rice. We can use potatoes. Okay, so we can use different type of raw materials to produce alcohol. But how we produce the alcohol? Basically, we use microorganisms. So you can ask me, cannot we do it by using chemical? Because say we are using grape to produce alcohol. What basically we do? I mean, what is need to do here? Grape has sugar. So we need to change that sugar into alcohol. So theoretically, we can do it by, chemically. Theoretically, it's possible, right? It's just chemical changes. So theoretically, it is possible. But what will be the problem if we just try to use the chemically? If we try to convert sugar into alcohol chemically, the process will be very complex. It will be very costly. Okay. But here's come the microorganisms. There are some microbes who can convert the sugar content of those raw materials, say like the sugar of the grape, into alcohol very efficiently. Okay, so that's why we use microbes to do this job. We use microorganisms to produce alcohol from different type of raw materials. Okay, alcohol is a very big industry, not only as beverages. Alcohol has many other industrial use. It is used in the pharmaceutical industry. It is used in alcohol have industrial use. It is used as a solvent in many industry. Okay. Vinegar production, citric acid production, mushroom. So you know what vinegar is, right? Sirka, we all
Yes,
use vinegar.
Yes, yes, sir.
Vinegar is what? Acetic acid?
Ethanoic acid.
I forget. Can you Google it?
Yes, sir.
What is vinegar? Acetic acid, right? Or what it is?
I think it's ethanoic. Yes, sir. Ethanoic acid. Acetic acid or ethanoic acid.
Okay, acetic acid or ethanoic acid. So, I mean, in Bangladesh, we can buy the vinegar in the market, right?
Yes,
Very
sir.
cheap, and the price is very cheap. So, usually these are, you can say, like cheap acid, cheap vinegar. I mean, these are just a diluted acetic acid. I mean, that acetic acid is produced by some cheap means, less costly industrial process, and they just dilute in the water. But originally, okay, sorry, where is the PowerPoint slide? Okay, but originally, vinegar is produced from like grape or rice. So first, it is a two-step process. First, say for example, you want to produce vinegar from rice. So first, you need to produce alcohol from that rice, and then from that alcohol, you have to produce vinegar. And in both this step, microorganisms are used. Okay. So that kind of vinegar are costlier. If you go to the, I think, supermarket, you will find different kind of vinegars, rice vinegars, grape vinegars, etc., etc. তোমরা এটা দেখছো নাকি সুপারমার্কেটে কখনো? অরিজিনালি আমরা যে ভিনেগার গুলো মার্কেট থেকে কিনি, এগুলো তো দাম খুবই কম মনে হয় না, 100 টাকার কম হয়তো বা around 100 টাকা।
Yes, sir.
এগুলো কিন্তু সস্তা ভিনেগার আর কি। হ্যাঁ, জেনুইনলি অরিজিনাল ভিনেগার তৈরি করা হয় ইয়া থেকে, মানে ওই ডাবল ফার্মেন্টেশন করতে হয়। ধরো তুমি চাইলে গ্রেপ থেকেও বানাতে পারো। গ্রেপ থেকে প্রথমে ওয়াইন বানাবা, পরে ওয়াইন থেকে ভিনেগার বানাবা। এই জন্য কিন্তু ভিনেগারের মানে কি বলা যায়, যে জেনুইন ভিনেগার যেটা, সেটার প্রাইস কিন্তু ওই অ্যালকোহলের থেকে বেশি হয়, অ্যালকোহলিক বেভারেজের থেকেও। এবং ওই ভিনেগার গুলোর একটা স্মেল থাকে আর কি। তুমি যদি গ্রেপ থেকে বানাও, একটা গ্রেপিশ স্মেল থাকবে, রাইস থেকে বানালে একটা রাইস টাইপ স্মেল থাকবে আর কি। এনিওয়ে, so these are some of the use of microbes in different food and beverage production. Then some microbes are directly used as animal feed. Okay, so for example, some Saccharomyces, they can be used as poultry feed. Okay, and Pyrococcus, it's a bacteria. It can be used as a poultry feed. And not only this, I mean, some microbes, they're directly used as feed, animal feed. But some microbes, they are used to process animal feed. Process animal feed, say, for example, there are many raw materials which are not easy for the like animal or chicken to digest. So we can use microbes to do some digestion. Okay. So say, for example, a very complex carbohydrate, so it's difficult for the chicken to digest. So we can use microbes to make that complex, to convert that complex carbohydrate into less complex carbohydrate, so that the chicken can easily consume it and digest it. Okay. প্রসেস করার জন্য আমরা ইউজ করি কিছু মাইক্রোবস, এরকম ইউজ করা হয় যে অ্যানিমাল ফিডটাকে একটু প্রসেস করার জন্য, যাতে সেটা হচ্ছে ওই অ্যানিমালের জন্য ইজি টু ডাইজেস্ট হয়। ওকে। আবার কিছু মাইক্রোবস আমরা সরাসরি তাদের মানে সরাসরি হচ্ছে খাবার হিসেবে ইউজ করা হয় পোল্ট্রি ফিড, যেমন পাইরোকক্কাস। পাইরোকক্কাস ইজ আ ব্যাকটেরিয়া and itself can be used as a poultry feed. Then in agriculture, I think you know the some of the use of microbes in agriculture. I mean, microbes are very important, necessary, I mean, essential to maintain the soil fertility, because they decompose the dead body, and while doing so, I mean, while decomposing the dead body, they release many important chemicals such as amines, sugars, vitamins, minerals, okay, fatty acids, etc., etc., which help to increase the soil fertility. Okay, and there are some microbes which help to produce organic nitrogen compounds. I mean, nitrogen is present in the air. It's a gas, so it's present in the air, but for trees, for plants, it's difficult to use this inorganic nitrogen or elementary nitrogen which is present in the air. So what some microbes do, they can use this elementary nitrogen and convert them into organic nitrogen compounds. And these organic nitrogen compounds, I mean, then the plant can easily use this organic nitrogen compounds. Okay. And this kind of bacteria, usually they form a relationship with the plant. I mean, they live within the plant or in very close vicinity with the plant, and they do this conversion and make the inorganic nitrogen available for the plant. Okay. Some microbes, they have insecticidal activity. Say, for example, Bacillus thuringiensis, it's a bacteria. It's produce a chemical which has insecticide activity. I mean, it can kill harmful insects. So Bacillus thuringiensis, you can say it's a biopesticide, bioinsecticide. Okay, it is commercially used, Bacillus thuringiensis. In industry, ethanol production, various enzyme production. Enzymes have many use in the industry. Okay, many use. In the what can I say, like in the dyeing industry, dyeing মানে ওই যে কাপড় চোপড় রং করে না, in the dyeing industry, in the washing industry, and also in preparing, say for example, in preparation of many food, many enzymes are required. Okay, so many enzymes are commercially produced by using microbes. Okay, in detail. Okay. And okay, yeah, some microbes they are also used in the mines, খনিতে আর কি, হ্যাঁ, যে for metal processing. I mean, এই আমরা যে বিভিন্ন মেটাল কোত্থেকে পাই, খনি থেকে পাই না? এই খনি থেকে তো পিওর ফর্মে থাকে না আর কি, হ্যাঁ। সো অনেক মাইক্রোবস আছে যে মাইক্রোবস গুলো ইউজ করে আমরা খনি থেকে মেটাল গুলাকে এক্সট্রাক্ট করতে পারি। ওকে, we can produce gas, methane gas, by using Methanococcus bacteria. Do you guys remember the biogas plant? We can produce gas from by using like cow dung or waste.
Yes, sir.
গরুর গোবর থেকে গ্যাস উৎপাদন করা হয় না, বায়োগ্যাস?
Yes.
Yes, sir.
Yes.
ওকে, সো যে আমরা দেখছি না যে কি হৃদয়ে মাটি মানুষের একটা অনুষ্ঠান ছিল না? There was a program aired in BTV. I mean, nowadays maybe it's aired in the channel I. But previously
Yes, sir.
there was no private channel in Bangladesh. There was only BTV. Okay, okay. BTV was not a Batabi Lebu that time. Okay. Anyway, it was a joke. Okay.
Yes, sir.
আমার মনে হয় তোমরা বুঝতে পারছো কি বলছি, হ্যাঁ? ওই সময় বিটিভি আসলে বাতাবি লেবু টাইপ ছিল না। বিটিভি was a very good channel at that time, and that program, even it's the হৃদয় মাটির মানুষ, it did some really good thing at that time. I mean, there was no idea about the biogas things at that time. I mean, he introduced this in Bangladesh and make it famous. So, I mean, many places in Bangladesh, this plant was established at that time, and people were using cow dung to produce methane gas, which they can use for cooking or like producing electricity. So, who do this thing? Who produce this gas, which is mainly methane from the cow dung? Bacteria. Okay. So you can see microbes are very good, right? They are our good friend. Only few microbes are our enemy, but most of the microbes, they are our friend. Okay, then in health, in sanitation, wastewater treatment, pollution control, we can use microbes to deal with the increasing pollution of this universe. Say, for example, chlorinated pesticides. I mean, many pesticides which are used in the agricultural field, they contain many different types of very harmful chemicals. So, for example, chlorinated pesticides, they have high amount of chlorine. These are bad for the environment. Okay, so we have to use the pesticides for our agriculture, right, for cultivation. But the remaining of the pesticides, they will be harmful for the environment. Okay, they can mix with the water bodies and harm the organisms of the water body. They can harm the fish or a small whatever fishes like things. Okay, so in short, these pesticides are harmful for the environment. So how we can get rid of this? By using microbes. So say, for example, pseudomonas and alkanogens. These microbes, they can digest the chlorinated pesticides. Okay, so by this they can help in reducing the pollution. Okay, and petroleum spill, it's a bad thing, sometimes happened in the sea. মাঝে মাঝে যে জাহাজ থেকে যে তেল লিক হয় আর কি, পেট্রোলিয়াম স্পিল হয়, দেখছি না আমরাও যে অ্যাক্সিডেন্ট হয়ে জাহাজ উল্টা পাল্টায় গেল, তারপর পুরো সাগরময় তেল 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 হয়ে গেল। দিস ইজ ভেরি ব্যাড, রাইট? কারণ ওটা হচ্ছে গিয়ে বায়োডাইভার্সিটি নষ্ট করে সাগরের আর কি। And it's very difficult to remove the spilled oil from the sea by using the conventional method. Okay, so if we can use, okay, till now the use of microbes for this kind of purpose is not that efficient till now, but scientists from all over the world are trying hard to find microbes, to use microbes to remove this kind of petroleum spill. Okay, actually, I mean, this is just one example. Okay, scientists around the world, they are trying to find different microbes to deal with different types of pollutions. Okay, so for example, you know that the air of Dhaka city is very contaminated, right? It's very toxic. It has very high. I mean, our Dhaka city air has high amount of lead, right? It's a very high amount of lead, na?
Yes, sir.
Yeah, exactly.
সবকিছু
So,
আছে।
হ্যাঁ কি বললা?
স্যার সবকিছুই আছে ঢাকার এয়ারে।
এইটা একটা ভালো কথা বলছো। হ্যাঁ, এমন কিছু নাই যেটা ঢাকার এলাকায় পাওয়া যায় না। আমরা খুবই আনহেলদি একটা কন্ডিশনে বসবাস করি। আচ্ছা, যাই হোক, সে there are lots high amount of lead in the air, and lead is a very toxic chemical. So now, if you can develop, you can find a microorganisms, or you can improve a microorganisms which can take the lead from the air and digest it. It will solve our problem, right?
Yes, sir.
So we can do these kind of things. I mean, at least we can try. In fact, microbiologists are trying to do that. And so the global warming. What is the main culprit for global? Not the culprit. Which gas contribute to the global warming actually? Which gas?
So carbon dioxide and methane, these are
Yeah,
greenhouse gases,
yeah,
basically.
exactly. Carbon dioxide. So, say for example, if you can improve a microorganism or find a microorganism which can utilize the carbon dioxide, it will solve our problem, right? Well, there was one paper maybe last year or the last year. I mean, some guys they were able to identify or improve a microbes which can utilize their carbon dioxide from the environment. It is still many more works, research are required to find more efficient microbes or make a efficient process to remove the environment from the remove the carbon dioxide from the environment. Okay. Environmental microbiology is actually a good field. Though I don't belong to that field, but সবাই তো সব লাইনে যাবে না, but it's a good field. Okay. মানে especially in Bangladesh, most of the microbiology students they always think about the medical side, medical sector. They always think about the disease, pathogens. But you can see it has lots of many other applications, right? So all the microbiologists should not go for only one field. I mean, not only for the medical microbiology. Okay, you guys should think about also about the other fields. Medical microbiology is not the only field. There are environmental microbiology, agricultural microbiology. Okay. Okay, agricultural microbiology, those fields are dominated by agriculture university graduate, but that doesn't mean you cannot work there. Especially pollution. Okay, pollution is a very big problem for the university, right? Industrial microbiology. You guys also should think about this field. Okay, not only think about the disease thing. There are lots of people to work with the disease. Okay, I'm not discouraging you that you cannot go for that, but I mean there are lots of other area where you can work in future. মানে আমি এটা আমার পার্সোনাল এক্সপিরিয়েন্স থেকে বলতেছি, বাংলাদেশের বেশিরভাগ এই মাইক্রোবায়োলজিস্টরা তারা হচ্ছে শুধু মেডিকেল লাইনে যেতে চায়, বাট ওটা তো একটা পার্ট। তো আমাদের আদার লাইনগুলো নিয়েও চিন্তা করা উচিত। ওকে, মানে বেসিক্যালি চিন্তা করা উচিত কি তোমার যেই লাইন ভালো লাগে তুমি সেই লাইনে যাবা ফিউচারে। মানুষজনের কথাবার্তা শুনতে হয় না। If you like environment, go for environment. If you like industry, go for industry. If you like medical, go for medical. Listen to your heart, not to others. Every field has job scope, job opportunities globally. Okay, so don't listen to other people, listen to your heart, trust me. I mean, and unfortunately in Bangladesh, okay, in Bangladesh there are quite good number of job for microbiologists, but if you think globally, all this field has job and work opportunities there. Okay, so it doesn't mean that if you the environmental microbiology is most demanded, or it has the most job scopes, or the medical microbiology is the most demanded. It's not like that. Okay, so every area there are work scopes. There are scopes for job and do research or work. Okay. Okay, then finally medicine and therapy.
Microbio has a bigger field abroad, right? যেমন বাংলাদেশে তো তেমন মানে মাইক্রোবায়ো নিয়ে
ওকে, বাংলাদেশে মাইক্রোবায়োর জব আছে, ভালোই জব আছে, কম না, হ্যাঁ। Just say for example, there are government research institutions, they have fixed post for microbiologists. All the government institutions, like say, I mean, I'm not okay. If we just exclude the universities, because of many public universities or private universities, they have faculty positions for microbiologists. Okay, I'm excluding those. Those are very limited numbers. Okay, but the research institutions, say government research institution, BCSIR, NIB, or Atomic Energy Commission, they have microbiologists, post for microbiologists. ICDDRB. Okay, but well, the required number are few here, but especially in the pharmaceutical industry, our pharmaceutical industry is booming. I think you guys know that our pharmaceutical industry is really booming, because most of our pharmaceuticals, they export medicine, and this is increasing every year. And all the pharmaceutical companies, they have a separate microbiology department. Okay, so you can work there as a microbiologist, and not only microbiology department, the pharmaceutical industry, they have R&D department, research and development. You can work there. Okay, but in abroad, I mean, it was about Bangladesh, but in abroad, okay, there are lots of work scope. Okay, lots of work scope. এত ডিসহার্টেন হওয়ার কিছু নেই। মাইক্রোবায়োলজি একটা ব্রড সাবজেক্ট, এর অনেক অ্যাপ্লিকেশন আছে, এবং কোভিড-19 আমাদের মানে মন্দের ভালো, এটা কিছু কিছু দিক দিয়ে একটু উপকারী করেছে, হ্যাঁ। I mean, people, especially in the developed world, the infectious disease thing. I mean, infectious disease is more prevalent in underdeveloping countries or the developing countries like Bangladesh or this kind of countries. In the developed countries, infectious disease is not that how can I say, prominent or frequent. Okay, so people of those countries used to microbiology was getting less preferences. I mean, but people should not do that, especially new diseases. We call it emerging and re-emerging diseases. New diseases are coming more frequently nowadays. এখন কিন্তু খুব ঘন ঘন প্যান্ডেমিক হয়, প্রতি 10 বছরই একটা করে প্যান্ডেমিক হয়, এমন অবস্থা। আগে কিন্তু এত দ্রুত হতো না। The bacteria, they are not only bacteria, all the microbes, they are getting resistant. So microbiology, this subject will again get its preferences there. And especially this COVID-19 pandemic, it shows us that, okay, microbiology is not done. Okay, there are a lot more things to do in microbiology. একদিক দিয়ে আমাদের জন্য একটা উপকার করছে আর কি, লোকজন এই মানে অনেক দেশেই এই মাইক্রোবস এর গুরুত্ব কমে যাচ্ছিল আর কি, সে গুরুত্বটা আবার বেড়ে গেছে। সব খারাপের মাঝে মাঝে কিছু ভালো দিকও আছে আর কি। এটা আমি না ঠিক ফানি স্টাইলে বললেও এটা একটা সিরিয়াস ইস্যু। ড্রাগ রেজিস্ট্যান্স, এটা একটা সিভিয়ার প্রবলেম হয়ে দাঁড়াতে যাচ্ছে। অ্যান্টিব্যাকটেরিয়াল রেজিস্ট্যান্স, okay, it's going to be a very alarming issue. It's already an alarming issue, and it's going to be more alarming.
স্যার এই অ্যান্টিব্যাকটেরিয়াল রেজিস্ট্যান্সের জন্য সায়েন্টিস্টরা একটা নতুন মেথড ডেভেলপ করছেন ফেজেস।
কিসের জন্য?
ব্যাকটেরিওফেজ দিয়ে ব্যাকটেরিয়াকে কমব্যাট করা ইউজিং ব্যাকটেরিওফেজেস।
আচ্ছা, আমার একটা কল আসছে, হ্যাঁ, এক সেকেন্ড আমি একটু ধরি। জাস্ট ওয়ান সেকেন্ড। হ্যালো, ওয়ালাইকুম আসসালাম। এই বাবা, আমি যে ক্লাস শেষে ফোন দেই। আধা ঘন্টা পর ফোন দাও। ওকে, ইয়া। ওকে, সরি, আই এম ব্যাক। হ্যাঁ, কি জানি বলতেছিল তুমি?
স্যার আপনি যে বলতেছিলেন ব্যাকটেরিয়াল রেজিস্ট্যান্স ডেভেলপ করতেছে ব্যাকটেরিয়া, তো আমি একটা ভিডিও দেখছিলাম যে যখন ব্যাকটেরিয়া আমাদের অ্যান্টিবায়োটিক্স এর রেজিস্ট্যান্স ডেভেলপ করে, দে লুজ দ্য অ্যাবিলিটি টু রেজিস্ট ব্যাকটেরিওফেজ। সো এখন এটা হচ্ছে একটা নতুন ওয়ে অফ কমব্যাটিং অ্যান্টিবায়োটিক
তুমি
রেজিস্ট্যান্স।
কি ওই যে কি জানি বললো, ভাইরাস ইউজ করার ব্যাপারটা বললা?
ব্যাকটেরিওফেজ।
Bacteriophage. Okay, so bacteriophage is a virus. Yeah. You'll learn about the bacteriophage in this course, in this MIC 101 course later. We have separate chapter on that. Bacteriophage are virus. Those virus, they infect bacteria and kill the bacteria. Okay. So people are trying to use bacteriophage as drug, as antibacterial therapeutics. Okay.
আর স্যার এটা কি কাজ করতেছে? Is it working?
Not for all the bacteria, because the thing is, first you need to find okay, bacteriophages are bacteria specific.
Yes, sir.
Okay, so for example, antibiotics, most of the time, I mean, the antibiotics, they are not species specific, right? They kill different type of bacteria, but bacteriophages, they are species specific, not only genus, they are species specific. Okay, so first, we need to find out efficient bacteriophages which can be used, which can be clinically used again the pathogen. Okay, so people are working on that. I mean, in last, maybe not in last year, in 2018, one group of scientists, they successfully used bacteriophages against drug-resistant bacteria in a kid, I think. Yeah, maybe she was like a 13 or 14. I forget her age. In USA, they were successful in using. I mean, that kid. I mean, she was diagnosed with superbug microorganisms. Superbug microorganisms means, I mean, those pathogens were resistant against all the available antibiotics. Okay, so as a last try, I mean, she was dying actually, but as a last try, they tried the bacteriophage, and alhamdulillah, they were successful. Okay, so people are trying. Okay, so people are trying, but some people, they're trying to invent new drug, new antibiotics. Some scientists, they're trying to find alternate ways. Bacteriophage is one of them. Okay, সো এগুলা না কাজ করতেছে অনেকে, হ্যাঁ, এবং এটাই আসলে সায়েন্স। অনেক ধরনের মানে অ্যাপ্রোচ আছে, ডিফারেন্ট সায়েন্টিস্টরা ডিফারেন্ট অ্যাপ্রোচ নিয়ে কাজ করতেছে। সবগুলোরই দরকার আছে, হ্যাঁ, সব ধরনের অ্যাপ্রোচেরই দরকার আছে। বুঝতে পারছো?
জি স্যার।
Okay, by the way, this last slide, medicine and therapy, it shows some use of the bacteria for the medicine and therapeutic use. Say, for example, insulin. You all know what insulin is, right?
Yes, sir.
Yes, sir.
Insulin can use for a diabetes patient. I mean, why diabetes patient they use insulin?
Sir, blood glucose concentration কমানোর জন্য।
Exactly. Because the diabetes patient, they can't produce insulin, so an insulin is necessary to take up the glucose, to uptake the glucose from the blood. I mean, when we eat something, eat something means we don't eat anything, right? We eat food. So when we eat food, we digest that food, and from the carbohydrate we produce sugar, and that sugar circulate through all over our body through blood. Blood হচ্ছে আমাদের পুরো বডিতে এই গ্লুকোজটা সার্কুলেট করে, and then different cells or tissues of our body, they uptake the glucose from the blood. And for this, insulin, it's a hormone, insulin is required. So if we don't have insulin in our body, then our cell will not be able to uptake the glucose from the blood, and it will cause the diabetes. Excess amount of glucose in the blood is very dangerous for our body. Okay. So, and there are lots of diabetes patients around the world, and they need huge amounts of insulin every year. Previously, I mean, in the beginning, and you know, I mean, who produce the insulin? Bacteria don't produce the insulin. Okay, they don't have diabetics. Bacteria, the diabetes হয় না, তাদের insulin produce করার দরকার নেই. Okay, so they don't produce insulin, but there are lots of diabetes patients in the world. So how the industry is going to produce this amount of insulin? In the beginning, okay, I forget the exact year, so maybe in around 1960s or 1970s, insulin was harvested from fecal calf serum. গরুর বাছুর আছে না, বাছুরের সেরাম থেকে সব এটা কালেক্ট করা হতো আর কি। ওকে, মেবি ওকে, সো মানে অ্যানিমেল লিভার থেকে এটা কালেক্ট করা হতো আর কি, প্যানক্রিয়াস থেকে বা সেরাম থেকে কালেক্ট করা যায় আর কি। হ্যাঁ, কারণ এগুলো সেরামে থাকে তো। বাট how many animal you are going to use to harvest that huge amount of insulin? It was very inefficient. Okay, so it was not practically, it was impossible, right?
আর ethical reasons ছিল স্যার, ethical reasons এর জন্য।
এথিক্যাল রিজনস ছিল, ধরো এথিক্যাল রিজনের থেকে বড় রিজন কি, ওই অ্যানিমেলের থেকে না খুব বেশি তো পাওয়া সম্ভব না। তুমি আর কতটুকু পাবা? এথিক্যাল রিজন একটা আছে, কতটুকু পাবা। এখানে আরেকটা দেয়ার ইজ অ্যানাদার রিস্কি থিং, দ্যাট ইজ, I mean, it's not good to, I mean, use something from other animal. There is a high chance of disease transmission. But anyway, it was not practically possible. So here's come the microorganisms. By using genetic engineering, okay, more specifically, it is called recombinant DNA technology. Okay, what is recombinant DNA technology? We'll learn it in later courses, so don't worry about it. For now, just remember the name. Genetic engineering technique, more specifically called recombinant DNA technology. By using that technology, scientists were able to modify bacteria. Say, for example, E. coli. They were able to modify E. coli to produce insulin. E. coli don't produce insulin, okay, but we can modify the E. coli genetically so that they can produce insulin. Okay, so using this genetically modified E. coli, we can produce huge amount of insulin by using fermentation. Okay, so whenever we want to produce large amount of a desired product by using microorganisms, we need to do fermentation. Because okay, fermentation, basically fermentation, I mean, by fermentation we can grow huge amount of microbes. Okay, we can theoretically, I mean, we can grow microbes in the laboratory by using culture media, but it will not be efficient. Okay, we will not be able to produce that high amount of microbes. So that's why it is necessary that we use fermentation. Okay, anyway, আমরা যেরকম জেনেটিক্যালি মডিফাইড মাইক্রোঅর্গানিজম ইউজ করে তাদের ওই ইউজ করে আমাদের ডিজায়ার্ড প্রোডাক্ট তৈরি করতে পারি। So theoretically, it is possible to produce any product by using microbes. But first, we need to modify, I mean, we need to genetically modify microbes to do so. Okay. Theoretically possible, practically সবসময় পসিবল হয় না। হ্যাঁ, প্র্যাকটিক্যালি কাজ করতে গেলে অনেক ধরনের কমপ্লেক্সিটি তৈরি চলে আসে। এটা যখন নিজেরা ফিউচারে কাজ করবো তখন বুঝবো আর কি। হ্যাঁ, but people are trying. I mean, many products currently are produced by using microbes, like medicines, like insulin-like things, drugs, enzymes, different chemicals, alcohols. But it's still, scientists are trying to improve or identify or develop or modify microbes to produce different, I mean, many other different important commercially or medically important chemicals. Okay, এগুলো একটু পড়ো আর কি। হ্যাঁ, পড়লেই বুঝবো, সহজ জিনিস। এখানে কিন্তু জাস্ট অল্প কিছু ব্যাপার দেওয়া হয়েছে। তোমরা যদি ইয়াটা দেখো টর্টরা বই, I mean if you look the torture chapter, I mean torture book, you will see lots, I mean many other applications of microbes. Okay, I suggest that just try to give a look. It will how can I say, it will motivate you. I mean maybe you will find more interest in microbiology.
So আমরা কি আজকে লেকচার 5 করব?
লেকচার 5 আমরা আজকে আর পড়বো না, সময় নাই।
তো এটা
But-
নেক্সট ক্লাসে
But the thing is, okay, please, okay, lecture 5 and lecture 6, these two are old thing. I think you have already studied this in your higher secondary. These are about prokaryotic cell and eukaryotic cells. Most of the topics are old topics, so please have a look. Okay, I want to finish both lecture 5 and 6 in the next class. Okay,
ওকে স্যার।
স্যার
Okay, sir.
again, I mean I don't want to waste too many times with these lectures because these are old things. Okay, there are other important topics like bacteria, virus, okay, disease pathogenesis. I want to use more time for those courses. Okay, so please study the lecture 5 and 6, and in the next class ask me question. Okay,
Okay, sir.
Okay, sir.
okay.
Okay, sir.
Do you have any question
No,
or
sir.
anything
Yes,
you
sir.
want to discuss?
স্যার মিড টার্মের সিলেবাস কি হবে?
Yes, sir. Syllabus কি কতটুকু?
আচ্ছা, মিড টার্মের সিলেবাস নিয়ে তো কোনো চিন্তা করি নাই। দাঁড়াও। Okay, so most probably the midterm syllabus will be like until lecture 6 or 7. Lecture 7.
7টা লেকচার স্যার।
It will be open book exam, don't worry. The good part of online learning is you don't have to memorize anything. You just have to understand the things. If you
Yes, sir.
understand the things, you can easily answer the question.
স্যার এটা যদি অন্য কোর্সে অ্যালাউ করতে তাহলে আরো ভালো হতো।
সব
Thank you,
কোর্সে
sir.
তো অ্যালাউ করে, সব কোর্সে তো ওপেন বুক এক্সাম নাকি না?
না স্যার, মাঝে মাঝে ঝামেলা করে।